Hi everyone, today we will be learning about developmental networks and gene regulation. Before we begin, we must understand how developmental networks operate on a basic level. First, we must know that RNA polymerase II has a hard time binding to the core promoter region of DNA without the help of particular transcription factors called activator proteins. Activator proteins bind upstream of the core promoter and help RNA polymerase II bind to the promoter, enhancing transcription. The mRNA created undergoes processing before being transcribed into a functional protein. However, there are also transcription factors known as repressor proteins. Repressor proteins are a type of transcription factor that inhibit the binding of mRNA polymerase II to the promoter. This prevents transcription and therefore results in no protein synthesis. Now we can put these ideas together. Since transcription factors are proteins, they are the gene products of other genes. A developmental network showcases the relationship between genes that encode other genes' transcription factors. The main idea is that the multiple gene products interact to produce a phenotype. We can see here that a activator protein produced from some other gene comes in and helps RNA polymerase II create a gene product, which is then used the repressor protein of this subsequent gene. We will use an example from The Lion King to explain this concept more clearly. In this example, characters of the gene and the effect characters have on other characters represents the kind of regulatory protein the gene produces. The end phenotype is who becomes king. Here we have the basic analogy. In The Lion King, the wildebeest stampede kills Mufasa. Had Mufasa not died, he would have raised Simba to be king. However, since Mufasa was not there to guide Simba, Simba did not initially become a king. This allowed Scar to become king of the pride. Now, I will re-explain this concept using the wild-type developmental network. Here, the wildebeest gene produces a repressor protein that decreases transcription of the Mufasa gene. Therefore, the Simba gene is not transcribed because it cannot be promoted by the Mufasa gene products. As a result, the transcription of the Scar gene is not inhibited since there are no proteins to block this transcription. This results in the wild-type phenotype of Scar becoming king. Here is an example of a single mutation in the developmental network. I will first explain this in the movie's context. Had there never been a wildebeest stampede, Mufasa would have lived to raise Simba to be king. Simba would have prevented Scar from ever being king. The single mutation developmental network works similarly. If there are no repressors produced to repress transcription of the Mufasa gene because the wildebeest gene is mutated, then the transcription of the Simba gene is promoted by Mufasa's activator protein gene products. This allows the Simba gene products to inhibit the transcription of the Scar gene. Since a mutation has occurred, the end phenotype of the network probably changed too. Here, let's say that Simba becomes king instead, like he would have in the movie. Here is an example of two mutations in two different genes in the developmental network at the same time. If both the wildebeest gene and the Mufasa gene are mutated, then there is no way that the Simba gene produces repressor proteins that inhibit the Scar gene's transcription. As a result, the Scar gene is transcribed and Scar becomes king. In the context of the movie, the mutation in the Mufasa gene could represent Mufasa never giving birth to Simba. If there was no Simba, the wildebeest stampede never would have killed Mufasa, yet Scar would have still eventually become king because Mufasa would not have had a child to pass the throne to. Of course, mutations happen independently of one another, but hopefully this story helped you understand how these two mutations affected the developmental network. Thank you for watching. I hope you better understand how developmental networks work.